Tyrosine kinase movement. First, two signal molecules attach to two separate monomer receptors. Once the signal molecules attach, the monomer receptors move closer together and connect with one another, forming a dimer. This process activates the tyrosine kinases on the dimer, causing each tyrosine to produce a phosphate from ATP, converting it back to ADP. Then, specific relay proteins bind to the phosphorylated tyrosine, which activates them. These active proteins then allow for a cellular response. Phosphorylation cascade. When a signal molecule binds to a receptor, an active relay molecule is released. This relay molecule then activates the first protein kinase. Then the first active protein kinase activates the second protein kinase by phosphorylating it through use of ATP. Then the second active protein kinase activates the third protein kinase with phosphorylation. After this, the second active protein kinase is deactivated by a loss of a phosphate group. Then the third protein kinase activates an inactive protein, which emits a cellular response. Then the third active protein kinase is deactivated by loss of a phosphate group. Intracellular response. First, the signal molecule enters through the cell membrane. Next, it attaches to the cortisol receptor and the chaperone protein in the cytoplasm. When the signal molecule and the cortisol receptor attach, the chaperone protein is released, allowing the receptor to change shape and pass through the membrane of the nucleus. The receptor and molecule then travel into the nucleus and transcribe a piece of DNA, creating mRNA. The mRNA is released into the cytoplasm and ribosomes attach, creating a specific protein. G-protein movement. The G-protein is inactive when GDP is attached and floats in the cytoplasm. When a signal molecule binds to the G-protein receptor in the plasma membrane, the receptor changes, allowing the inactive G-protein to bind to it. This activates the G-protein by changing GDP to GTP. Then the active G-protein binds to an enzyme, activating the enzyme. This emits a cellular response specific to that enzyme. The G-protein is then deactivated and moves back to its original position.